Hello, my name is Kaitlin Lowran. I'm a graduate student at Oakland University in Dr. Colin Wu's lab. I will be discussing the type of DNA damage caused by microgravity. The human genome receives damage from UV radiation, oxidative stress, and other environmental toxins. Some types of DNA damage include the formation of abasic sites, base pair mismatches, or unusual DNA structures that lead to genetic instability. Human cells have repair mechanisms to correct these lesions. Defects in DNA repair components are associated with the onset of increased risk of cancer, heart disease, and other genetic deformities. In space, the most prominent mutagen is UV radiation, which forms crosslinks in genomic DNA. The long-term consequences of space exploration to astronauts and their DNA repair mechanisms is still unknown. In this work, I will use a rotary cell culture system to simulate a low-gravity environment to study the types of DNA damage formed from exposure to microgravity. My experimental strategy is that HEC-293 cells are placed into the RCCS and then treated with increasing concentrations of hydrogen peroxide, which causes oxidative stress, bleomycin, which causes double-stranded DNA breaks, or camptothecin, which causes single-stranded DNA breaks for 24 hours. Single-cell electrophoresis, or common assay, was used to measure the amount of DNA damage in the cell. Post-treatment, cells are embedded in a microscope slide with low-melting agarose and then lysed. The slides are then placed in an electrophoresis chamber, and since DNA is negatively charged, it will migrate towards the positively charged electrode when an electric field is applied. The DNA is then stained with cyber gold and imaged by epifluorescence microscopy. In healthy cells, their nucleoids will appear round because the DNA will travel uniformly during electrophoresis. The presence of DNA damage will result in a comet tail. The extent of each type of DNA damage will be quantified using an image analysis software to determine the amount of DNA in the head versus tail and then graphed. For my results, we used two different variations of the COMET assay to measure the relative abundance of double-stranded breaks, which is a neutral assay, and single-stranded breaks, which is an alkaline assay. HEC cells exhibit a higher sensitivity to microgravity because there is an increase in the percentage of damage in the controls in microgravity compared to normal gravitational conditions, seen by these lines here. And then to determine which type of damage, double-stranded or single-stranded breaks, was more detrimental to the cell, we added bleomycin or camptothecin. There is an increase in single-stranded breaks not only in the control, but an increase in the treatment of camptothecin. Based on the current data, microgravity has a negative effect on HEC cells. For our future directions, we want to test using cardiomyocytes and retinal ganglion cells, not that the method has been successful. Additionally, we would like to study other forms of DNA damage, such as oxidative stress and DNA crosslinks. Also, we want to determine the impact of long-term exposure in microgravity to cells and what delayed effects of microgravity can be seen once the cells return to normal gravitational conditions. I'd like to thank Dr. Wu and my colleagues for their continuous support. I would also like to thank the MSGC for their support of this project. Thank you.